So the title, Cancer Research Made Faster. Who am I? My name is Aaron Lisman. I'm the lead software engineer on the cBio portal. I work at the Nicholas Schultz Bioinformatics Lab at the Center for Molecular Oncology at Memorial Sloan Kettering Hospital. That was a mouthful there. What is cBio portal? I'm going to get more into this later, but to start with, it's a data analysis and visualization application for exploring genomic and clinical data aggregated from research studies and the clinic. So MSK is one of the largest cancer hospitals in the world, and a lot of the data that comes from our patients goes into the portal, and this is true for many other hospitals around the country and the world. cBio portal originated in 2012 at MSK. It's open source, grant supported. There are many different institutions that help maintain it. Dana-Farber, MD Anderson, CHOP in Philadelphia, and it's used all over the world by tens of thousands of researchers and clinicians. So history, 1 year and 218 pull requests ago, I stood on a stage not unlike this, smaller than this one, at a ClickHouse meetup, and told everyone about a POC that we had done that showed that ClickHouse could be used to solve a lot of our performance problems. Just like the last presenter, we had a lot of analytical queries on data that we were keeping in in MySQL, in an OLTP database, and we were just doing the exact wrong thing technically, and someone realized that for doing analytical queries, we should be using an analytical database. And that's how we found ClickHouse. And this POC showed us that we were right, that we could have incredible performance gains. So now we're a year later, and the good news is we've had tremendous success. And I'm going to show you some metrics about that a little later on. And we are now in production on our internal portals. And then in the next few months, we're actually going to be pushing this out to all of the many people around the world that use cBio portal locally at their institutions. So I want to, because I think it's interesting, I want to get into a little bit of the biology that cBio portal concerns. It's actually, cBio portal is actually a bioinformatics tool. Bioinformatics is just data science applied to biological systems and biological data. And bioinformatics is used to draw conclusions that inform treatment decisions and spark medical research based on patterns that are found in the data. Cancer happens to be a disease that's very given to analytical analysis and to data analysis because it is driven by the information, nature's information, the DNA in our cells. And it's actually a set of diseases, and they have in common that they're driven by a wide variety of genetic mutations, so damage to the DNA of many different kinds caused by many different things, smoking, for example, or sun exposure. And all of these mutations conspire in various ways to interfere with the body's ability to control tissue growth, and that's why we get tumors. So how do we decode the mystery of cancer? Well, we have the relatively new ability to cheaply sequence DNA, to read the DNA and detect the tumor's mutations. Sequencing is almost standard of care now, but that was definitely not the case 10 or 15 years ago. So there's this torrent of data now that wasn't there even 10 or 15 years ago. And it's now very pertinent to research and the clinic. And so reading this data lets us peer into the root cause of cancer, and it suggests biological pathways that are involved and worthy of inquiry. And it helps us to target therapies at specific mutations. So because the mutations are the root cause, this allows us to come up with treatments and target them much more specifically and intelligently. And we're now seeing that more and more. Drugs that are actually prescribed for specific mutations that are detected in people's tumors. So this is just an example of a very simple basic example of the kind of bioinformatic analysis that might take place in the portal. So you would you have a big cohort of patients, and you divide them into two groups. TP53 is probably one of the most famous and important genes in cancer. And these are the two groups here. And red is the group that has a mutation in TP53, and blue doesn't have a mutation. And you see that this is very clinically relevant. This is a bit heavy, but this is a Kaplan-Meier curve, and it shows survival. So you can see that the folks with the TP53 alteration don't do as well. So you might say, okay, well, I want to go and look at what TP53 actually does. And it turns out that it's a gene which helps to regulate damage and repair damage to DNA. So if you lose this gene, then you're just going to rack up more mutations. So pretty interesting. There's a whole book about TP53. So this is just an example of a few different visualizations that we have in the portal. You can see here how different mutations in different genes overlap. And this is our what we call our study view, and this is the subject of all of our refactoring and ClickHouse implementation. And what this allows us to do is filter a cohort of patients by a whole bunch of different facets. So what kind of cancer they have, how many mutations are detected, their smoking history, whether they have a mutation in a certain gene. So cBio portal is very widely used, and this curve shows how it's been adopted since its birth, back in 2011, 2012. So it's just been this steady ramp up, and it's used, all over the world. And then this map here shows all of the, there are now actually hundreds of different institutions which are using this software. So again, it's open source, it's free, and they can install it and use it. And ClickHouse will be part of that. The real message is, in our world, performance in our application means more insight. So if our application is incredibly sluggish, people aren't going to want to use it. They're not going to want to test all these different hypotheses. If it's fast and responsive, they will. And I think it will lead directly to more insight, and speed up progress. So it's a great cause. So getting now to the technology, what is the scale of the data? The internal MSK portal has 140,000 patients. We have another portal. We manage another portal for the Genie Consortium that has 220,000 patients in it. The human genome has 22,000 genes in it. An example of just one assay that we do. This is when we detect what's going on. It's not quite sequencing, but RNA expression data has a read per gene per sample, so that gets you to 4 billion rows very quickly. And we ultimately want to make sure that the portal can support millions of samples. The portal, the technology stack of the portal was initially designed to handle, tens of thousands of patients. And the result is that it has become extremely slow. We have researchers who have to sometimes wait a minute, they click a button and have to wait a minute to see, the result of their filtering. So we knew that we had to do something about this. Well, this is the study view that I already kind of described here. So this, of course, as I said before, led us to OLAP. What were we doing wrong? Well, we were doing much too much work in our service layer, as the fellow that from RAMP said to. We were bringing giant data sets into memory just to filter and count them in Java. And we were, generally just exchanging performance for what I like to call developer ergonomics. It was just, sort of easier to do things that way, and people didn't realize that that it was very unperformant at scale. So we've spent really the last 6 months heads down, and we've rebuilt 20 endpoints that filter patients and samples. We built a denormalized schema in ClickHouse according to the needs of these endpoints, and we re-implemented filtering logic in SQL. So that was, the most work was just trying to figure out, what all this Java was doing, and then figuring out how to re-implement it in SQL. I'll get into a little more about what we did, but I want to show the result first. And so on the left, you're going to see the new study view, and you'll see that it's very responsive. Not quite instantaneous, but close. And I actually think that if we wanted to, we could get to practically instantaneous. And on the right now, you're going to see the legacy application. I could probably leave it there for the rest of the rest of the talk. But you get the picture. It's really, really, really slow. You'd be sitting there waiting for 40 seconds sometimes, and so we were very embarrassed as developers. Yeah, and this is just a chart. These are the endpoints that we did, and this is the legacy response time, and blue is the ClickHouse response time. So it was like at least 90% improvement. 10 times faster, I guess, is a better way to say it. So how do we go about doing this refactor? How much time do I have? I'm good. Okay. Well, we wanted to get right to business, proving that the optimization concept was viable, and learning how to write, learning how to re-implement all of this logic in SQL. So we just copied the MySQL schema whole into ClickHouse using a tool called Sling, and it doesn't really take that long. We don't actually have that much data compared to a lot of people. And then we derived normalized views. Denormalized views based on these underlying tables. We tried to use materialized views, but we found that there was a little bit of an issue with how they triggered when there were sort of complex joins, and so that's something that we need to look into a little bit more. And I would love people's input. The mantra of our refactoring effort was, do not return voluminous data to the web server. Keep it in the database. Process it in the database. And so that forces you to get creative and complicated with subqueries. So all of our business logic now is in the form of these kind of complicated, deeply nested SQL queries. And of course, we use MyBatis to kind of to format that, and that helps a lot in terms of making these queries readable and composable and modular. But we still find ourselves in the situation where a lot of times when you need to debug something, you just need the whole query, which is 5 nested SQLs, and MyBatis doesn't really help you figure out what's going on. And so you just have to get this giant SQL thing, and it's kind of hard to reason about and debug. And also, unit testing that is difficult. You in Java, you would unit test this, at sort of the function level, the method level, but it's not totally clear to us how to do that. With SQL or MyBatis. So we do it with integration testing. We actually have a database that we spin up in our integration tests, and we just write against that. But it strikes me as kind of unideal. So what's left for us to do? We've proven that the optimization works. It's in production, and then we still have about 2/3 of our application that we haven't touched, and we don't really want to have 2 databases going. Ideally, we just have one. We just have ClickHouse, and so the question is, how much do we have to refactor a lot of this other stuff, which isn't so obviously OLAP, oriented. Services that list things. It's just not analytical process. So what we're going to see is can we run our legacy endpoints or our legacy logic against ClickHouse using, ClickHouse has the MySQL interface. And that's kind of the next experiment we're going to do. And I suspect that it will be fine. And so, as I said, going all ClickHouse is very attractive to us. But there's some open questions. Do we denormalize everything in the ETL process so we forget about having the whole MySQL schema sitting in ClickHouse? If we decide to denormalize everything, how do we maintain the data integrity, which we sort of, we get out of the conventional relational schema, which also is very, easy to reason about. So I'm a little bit reluctant to kind of lose that. The other thing that is we have to figure out how to do complicated binning logic. So we allow the user to do a lot of configuration on the histograms that we show in the application, and we want them to be able to continue to be able to do that. And it doesn't seem like we can accomplish that using the built-in binning logic in ClickHouse. So I think we're probably going to have to do user-defined functions for that. So that's a subject that we're going to be exploring. So that's all I got. Thanks again. And thanks to ClickHouse.
Yeah, so now we're going to open the floor to any questions for Aaron. Just raise your hand. I'll point to you, and then Ryan will come over with the mic if you have a question.
A question about data ingestion. So did ClickHouse help at all in adding data to the database? Were you able to get it faster? Like how often does a database grow?
Yeah, so it depends what instance of the portal we're talking about. But the internal instance probably will be getting updated every day, but not every second, okay, or even every hour. So it's not that big a problem, we haven't solved that yet. And the data comes in the form of files, actually, files that are produced by these pipelines that handle all of this data coming out of the clinic and from curation teams. And so one thing I'm hopeful about is that I think ClickHouse has the ability to ingest from files. And so that could make our lives very easy.
Does ClickHouse have an on-prem solution for ingesting the data like you're mentioning? Uncurated?
I'm sorry, ClickHouse or cBio portal?
You said you wanted to submit unstructured data to ClickHouse? You said files, but
files. Yeah. I mean, they're structured in there's some structure in the files. They're some form of serialization. Yeah, I'm not quite sure how to answer the question.
Okay.
Sorry.
Two questions. When you said you didn't have a lot of data, is it gigs, tera, penta, how big? And then I'm not familiar with Sling. Are they slinging files, or what else are they slinging into ClickHouse?
Well, Sling can go from many different sources, but we're slinging from MySQL to ClickHouse. I actually don't know the answer to that. Yeah.
I actually have a question about the cBio portals. So do you know the Galaxy community? The Galaxy. Because previously I also did the data platform for support the bioinformatics. So I knew there about more than 10,000 tools and probably hundreds of workflows. So for cBio portal, I see you actually focus on the cancer research, probably focus on some of the workflows. So do you know the Galaxy community?
Yeah. The fellow sitting behind you probably would answer that question a lot better than I would.
You from Galaxy? Okay, okay. So what kind of what are your relationship between you two? So, are you are one of the tools or workflows in the Galaxy provided to open to the Galaxy community, or you are separate, totally separate?
I think we're totally separate. Yeah.
Let's try to answer that one. That's a very specific bioinformatics question. So, yeah, I mean,
Because I developed the data platform, but after I developed the platform, then I see you have the Galaxy. So.
right. Yeah. So they're quite different tools, and the Galaxy is really more for running workflows, and it has visualization tools built in as well, but it's really for sort of a platform to manage workflows and allow sort of easy UIs for people to stitch workflows together. Right. Yeah, cBio Portal is a bit more on sort of the data visualization, analytics dashboards side.
Galaxy, because the workflow, the tool or workflow, they could provide this function.
Right. Yes. Exactly. Yeah. So we are working with some of the people in that community to sort of integrate the viewer. Thanks.
Thanks for that. There's a question. There it is. Good job. Ah, traps.
Hi. So I have a question about cBio Portal. Is it modular? Can or you have a standard dashboard for all your researchers? Can they build custom dashboards for their specific needs? And how seamless is that with your overarching architecture, I suppose?
Yeah.
That's what I'm saying.
So we do have some ability to customize things at the kind of the tab level. And that's certainly an idea, a kind of an app store idea is something that we've entertained before. So, researchers may have some particular algorithm that they want to run or some visualization that they want to build. And, it would be great if the whole community could sort of use that too and leverage that. So it's something that we've certainly thought about. It's somewhat limited right now. Unless people want to, contribute directly as open source contributors, and we certainly do. We do work that way with a number of different entities, consulting companies that are building features on behalf of drug companies or hospitals.
I'm curious how the transition from MySQL to ClickHouse was from an engineering familiarity perspective. Obviously, it's still SQL at the end of the day, but
Yeah.
they're very different things under the hood, especially with a big open source project where you have a lot of folks contributing to it. How did you manage that?
Right. Well, I mean, I actually don't think we've used that much, ClickHouse specific SQL. We've certainly done, we certainly noticed that if you get the keys right, the primary keys right, your performance it really helps performance. So that's something that, and also getting the data types right. That took a little bit of understanding. But I mean, I would say that most of the difficult work was just learning how to, sort of translate these sort of, control flows, that are very sort of intuitive in imperative coding into SQL. One thing that really helped was, oh, sorry, if we can hear me. One thing that really helped was the with statement, so you don't have to actually physically nest all of these subqueries inside of each other. So it seems like such a simple thing, but that was very helpful. And then I mean, getting better at MyBatis. We've always used MyBatis, but we're using it at a much deeper level. Yeah.
Okay, awesome. Oh, can you guys hear me? Oh yeah. Okay, awesome.
Yeah,
And yeah, with that, thanks so much, Aaron. And we can get into our next speaker for the night.
Thanks a lot, everyone.